So this lecture is presented to you by DVL, which is extract of IDVL textbook. Okay, so dermatology video lectures. So first lecture of this video series is anatomy of skin. This lecture is presented to you by Dr. Udit Sinha, that is myself, with level degree holder in dermatology, venereology, and leprosy. So structure of skin. So the largest organ of human body. So what are there here? We have the epidermis, the dermis, the hypodermis, which is also known as the subcutaneous fat. Okay. So we have then this basement membrane zone, which is the junction between the epidermis and the dermis. So we will be dealing about this in later chapters. So first we will just focus on the anatomy of skin. So this is the skin. We have here the epidermis, the dermis, and the subcutaneous fat. The epidermis being extracted over here. You can see we have separated epidermis in this image. And then what are the parts of dermis then? And there is this papillary dermis. Then we have this peri and nexal dermis. These both together can also be called as adventitial dermis. Then we have reticular dermis, is just below the papillary dermis. Then we have subcutaneous fat layer. And here we have marked different structures. We have this retinatus which goes down. So here there is mnemonic रीटा नीचे जाती है. So रीटा goes down. So retinatus are the one. You always get confused between what are papillae and what are retinatus. So रीटा goes. Remember, retinatus comes from epidermis to dermis. Papillae, these are the papillary extension into the dermis. So papillae also have these capillary loops, superficial and subpapillary plexus of blood vessels. And we have the sebaceous glands, dermal duct of eccrine sweat gland, nectar pili, hair follicle, deep plexus of blood vessels. Okay, loose connective tissue of subcutaneous fat, and then lobules of adipose tissue. So we'll be discussing about all these in the coming chapters. For now, we'll be sticking to the same topic. Then we have the stratum corneum. What is stratum corneum? The topmost layer of the skin. Then we have the stratum lucidum. Stratum lucidum is the only layer which is present in the palms and soles. These are not present all over the body and all over the skin, but only present in the skin, in the palms and soles. Then we have stratum granulosum, which is the granular layer. Then we have stratum spinosum. Spine is the thickest layer of the epidermis. Then we have the stratum basale, and then we have the dermis below. So we'll try to correlate this image with the HNE. So what we have in HNE? So stratum corneum. So this is from corneum, and we have stratum lucidum, stratum granulosum, stratum spinosum, stratum basale. Now if you read here, you'll find it very easy to understand. But in exam, you get confused which layer is what. So now I'll give you a small trick. This the darkened layer is obviously what? The granular, because there are granules here. So this is darker layer. Okay. Then the layer above it is what you know already with the mnemonic. Okay, so let me tell you the mnemonic if you don't know. Come, let's get sunburn. So come, let's get sunburn. This corresponds to the first letter of all the layers of the skin. Okay, come, let's get sunburn. Okay, so the darker layer is granular layer. So what we have above G, we have L. So lucidum layer is just above the darker granular layer. So you know lucidum layer is here, and darker layer below we have spinosum layer. S, spinosum layer, the thickest layer. Okay, this is roughly lucidum layer like this. Okay, then we have corneum layer. Then we have dermis, and above dermis we have stratum basale. In basale you can see there are many cells you can see here, but in spinosum layer the cells are less in number. This is how to identify. Then we have epidermis, embryology. Third week, a single layer of glycogen filled cells is formed. In sixth week, there is periderm. This has been given in detail in IDVL. And I suggest you go and read the book too after listening to the class. Here I am trying to put only the important points which if you write in the exam will fetch you good marks. So we have sixth week we have periderm, epitracheal layer or germinal layer. 12th to 16th week many microvilli and intermediate layers are formed. And 10th week we have hemidesmosome and desmosomal proteins. 11th to 15th week keratinization of skin appendages. So what is keratinization? We'll be discussing about in coming chapters. So keratinization of skin appendages. Basically what keratinization is formation of keratin. Okay, what is keratin? This this is this is a type of cell. This is a type of cell. Uh, is a type of cell present in the skin. So we'll be talking about all these things. Okay, then in 24th we have Wernicke's caseosa sheds. In kids there is something called Wernicke's caseosa that sheds at 24 weeks. Okay, then this is a normal graphic representation, not required. I have just put for your memory purpose. So this is schematic representation of the epidermis. We have stratum corneum, granular layer, spinosum layer, basal layer, DJ. What layer I have missed here? Lucidum layer. Okay, which is present in palms and soles. So made of stratified squamous epithelium. So a continuous renewing structure and gives rise to appearance like solution not required. Structure of epidermis: stratum corneum, stratum lucidum, granulosum, spinosum, basal, dermis. So this is the youngest keratinocytes. This is the oldest keratinocytes. The after old what you get, you die. So we find here the dead keratinocytes. We have here living keratinocytes. We have here dendritic cells. What dendritic cells I'll let you know. We have here melanocytes. We have here tactile cells. Merkel, we have Misners, we have down, and then we have a sensory nerve endings. Okay. Then we have here two variations. So here is normal skin. Here we have normal skin. Normal skin. Here we have skin of eyelid. You can see here this is very thin, right? So what is this layer? This is the epidermal thickness. Here we have a thick epidermal layer. Here we have thin epidermal layer. So this is both eyelid and acral skin. Layers of stratum basal, continuous single layer, thick 2 to 3 layered in glabrous skin, hyperproliferative, has dark staining nucleus, dense cytoplasm, ribosomes and tonofilaments, vacuoles with melanosomes. Okay, so basal keratinocytes attached to BMZ via keratin filaments K5 and K14 at hemidesmosomes. Okay, at hemidesmosomes. Primary site for mitotically active cells. 3 types of cells: stem cells, ultimate division unlimited division capacity, transient amplifying cells, limited capacity of division, and then we have post-mitotic. There is no capacity of division. Okay, so normal transit time of basal cell to enter the stratum corneum is 14 days. Stratum spinosum 8 to 10 layers, polyhedral cells with round nucleus, upper spinous layer larger, more flattened lamellar granules. Okay, we have desmosomes, although provides mechanical coupling for the physiological communication and are also integral mediators of intercellular adhesions. So these are forming K1 to K, the keratinocytes K1 to K19, 20 we have. Okay, so keratinocytes, depending on keratinocytes also we get to know what function they are going to do. So contains large keratin filaments K1 that insert to desmosome peripherally. So there are specific function of keratinocyte that we will be dealing later. So first, desmosomes, symmetrical laminated structures that interacts with keratinocytes and provide stability. Desmosomes, although provide a mechanical coupling for the physiological communication, are also integral mediators of intercellular adhesions. When these structures are disrupted, disease can result. Okay, we'll talk about disease later. Disease resulting from disruption of desmosomal protein. Desmoglin 1, we have pemphigus foliaceous, stratum striate palmoplantar keratoderma, staphylococcus, scarlet skin syndrome, bullous impetigo, pemphigus vulgaris, autosomal recessive hypotrichosis with desimoglin 3. We have pemphigus vulgaris, first fa fa foliaceous fa से first ठीक है. 3 sounds like V. Okay, we have next, we have desimoglin 4. If you reverse it, is hypotrichosis. Okay, we have hypotrichosis. Then we have placoglobin. से क्या होगा? palmoplantar keratoderma with woolly hair and erythmogenic right ventricular cardiomyopathy. It is called a Naxos disease. Placoglobin se you can ask Naxos, Placo-Naxo. Okay, then we have placophyllin, ectodermal dysplasia, skin fragility syndrome. So here we have skin erosions, dystrophic nails, sparse hair, and painful palmoplantar keratoderma. Okay, placophyllin 1, placophyllin 2. What is placophyllin 2? To arrhythmogenic right ventricular cardiomyopathy, if not required for us. Okay, so this one is required. Placophyllin 1, ectodermal dysplasia or skin fragility syndrome, SFS. Okay, SFS. Okay, SFS. And remember this one, placophyllin 1, desmoplakin, lethal acantholytic epidermolysis bullosa, striate palmoplantar keratoderma. Okay, then we have Darius disease. This is very famously asked question. What is the other name of Darius disease? So very common sounding name, right? Keratosis follicularis. This is Darius disease. What happens? This mutation is SALCA2. Then you have Haley, benign chronic pemphigus. Okay, mutation in ATP2C1. First is 2 here, not 1. 2C1. ATP2C1. Okay. Lamellar granules are also formed in spinosum layer. These secretory organelles deliver precursor for stratum corneum lipids to intercellular spaces. So what is stratum granulosa? So named for the basophilic keratinocyte granules, porphyrin, keratin filaments, and loristrin. Okay, these three are the granules present here. 2 to 5 cells thick. Cytoplasm of keratinocytes and uppermost layer of spinous cells contains lamellated granules, membrane coating granules, or Orlan bodies. Okay, so understand the cytoplasm of keratinocytes and uppermost layer of spinous cells. We are not granular layer. This uppermost layer of spinous cells contains lamellated granules or membrane coated granules, also known as Orlan bodies. The prominent site for production of epidermal barrier component and protein that process this component. The conified layer starts to form this layer. Okay, the conified layers start to form in this layer. Okay, so profilagrin will give rise to filagrin. The profilagrin will give rise to filagrin and keratin, and which can also be helpful in formation of microfilaments. Okay, now we have the then filagrin is degraded into urocinin and pyrrolidone carboxylic acid. This contributes to hydration of stratum corneum and filter the UV rays. Loristrin, after release from the keratohyalin granules, binds to desmosomal structures and crosslinks to transglutaminase 3 and 1 to form conified cell envelope. Then this conified cell envelope is essential for TEWL. What is TEWL? Transepidermal water loss. Okay, now we have this here mutation in the gene disease caused. So we have mutation TGM1. What is TGM1? Mutation TGM1, we have lamellar ichthyosis. Mutation filagrin, ichthyosis vulgaris. Okay, lamellar ichthyosis is different than ichthyosis vulgaris. This is the most asked question, ichthyosis vulgaris. Then we have the loristrin abnormality. We have Vohwinkel syndrome, which is ichthyosis plus pseudoaneum disease, progressive symmetric keratodermia. Okay, Vo- Vo- Vohwinkel syndrome. Okay, this is very difficult to pronounce and not commonly asked, so not that important. But what I'd suggest is remember it is these two, TGM1 and filaggrin. In stratum corneum, outermost skin layer, 20 to 25 layers of condified cells, flattened cells, lost nuclei, and cytoplasmic organelles joined by desmosomes which break apart and cells are shed. Corneocytes contain soft keratin, high molecular mass, more than 60 kilodalton keratin stabilized by intermolecular disulfide bonds. Has lipid depleted proteins enriched in corneocytes, surrounded by continuous extracellular lipid matrix. Lipids released from the laminar body surrounded by these cells responsible for permeability of this layer. Okay. Hydroconium is very common, and I'm sure you must have studied. This is the first layer that you study even in your MBBS days, and in PG days you definitely know more about it. So I'm not going in depth. So in formalin fixed specimen, cytoplasm with cysteine disulfide bond shrinks and forms a shell along the membrane. Characteristic basket weave architecture. In fluorescent stain, orderly vertical stacks. Stratum lucidum, electron translucent layer present in palms and soles, which is thicker in between stratum corneum and granulosum, are still nucleated and referred to as transitional cells. Okay, electron translucent layer present in palms and soles only. Okay, in between, obviously, stratum corneum and granulosum are still nucleated because the layers in corneum are anucleated. He is mentioning it. These are still nucleated and referred to as transitional cells because after this the nucleus will be lost. What are keratinocytes? Ectodermal derivatives contains keratin in intermediate filaments, tonofilaments in the cytoplasm. So that keratin is basic, type 1, numbered from 1 to 8, acidic, type 2, numbered from 9 to 9. So 1 to 19, okay, 1 to 19 keratin are there, right? So till 8 we have basic, till 8 we have basic, from 9 we have acidic, till 19. Okay, each are co-expressed to form filament structured, hence called as obligate heteropolymers. Plays a role in biochemical and immune mechanism. So we'll study about this skin estuary lymphoid tissue salt. So keratinocyte culture are used for skin grafts, burns, other defects. In gene therapy, releasing GH corrects type 7 collagen abnormalities in recessive epidermolysis bullosa. Okay. And what is GH? Please don't ask me. You must have been knowing this. What is GH? So specialized structure and function. Superficial cellular cohesion. Keratohyalin granules increase in size, surround tonofilaments to form mature epidermal keratin. Meanwhile, profilagrin forms keratohyalin granules in stratum granulosum. So we study this again. Specialized structure and function. Superficial cellular cohesion. Superficial cellular cohesion. Keratohyalin granules increase in size, surround tonofilaments to form mature epidermal keratin. Okay. Keratohyalin granules increase in size. The keratohyalin granules increase in size to form mature keratin. Meanwhile, the profilagrin forms keratohyalin granules. Okay, keratohyalin granules in stratum granulosum. Okay, keratohyalin granules in stratum granulosum. Okay, and filagrin in stratum corneum, which acts as glue for keratohyalin granules. Profilagrin forms KH. KH forms keratin. Okay, then we have filagrin, stratum corneum, which acts as glue between these two keratins. Simple. Filagrin mutation, atopic dermatitis with asthma. Okay, ichthyosis vulgaris. We have already spoken about this. Then we have barrier function. Odlan granules fills up the intercellular spaces and prevent water loss. Contains neutral sugars, hydrolytic enzymes, free sterols. Stratum granulosum, free sterols from sugars. Okay. Stratum corneum, neutral lipids, free sterols, no sugars, and lipid is hydrophobic. Okay. Barrier function we are talking about. So we have Odlan bodies. Fills up the interstellar spaces and prevent water loss. TEWL is included. TW, transabdominal water loss, is prevented here. Contains neutral sugars. Okay, hydrolytic enzyme. That's why the water is not being leaked, because there are hydrolytic enzymes there. Then free sterols, the fat molecules. In stratum granulosum also we have free sterols and sugars. In stratum corneum we have neutral lipids, okay, free sterol, and no sugars. Okay, no sugars. In steroid sulfatase removes lipid from stratum corneum, which is known as desquamation. The keratinocyte sheds in the corneum layer, right? And that is done by steroid sulfatase, which desheds the or desquamates the keratinocytes from the corneum layer. So enzyme activity, lysosome, hydrolytic enzyme. What are the enzymes here? Acid phosphatase, aryl sulfatase, beta glycolysis. Present more in granular layer. Secondary lysosomes or phagolysosomes, lower epidermis, digest phagocytized melanocytes. Okay, so barrier function we have spoken about. Enzymatic activity we have seen. In lysosomes there are three enzymes A, B, ARB. Remember, acid phosphatase, aryl sulfatase, beta glycosidase. Then more in granular layer. Okay, this is enzyme. Then secondary lysosomes are there. One is normal lysosomes, secondary lysosomes or phagolysosomes. Then lower epidermis digest melanocytes, the melanin producing cells. Okay. Then immune functions. We have production of interleukin alpha, 1 beta or 6 8. IL-1 alpha, beta, 6 and 8. Okay, wait 1 second. I'll again draw this. There is more confusion over here. Okay, let's erase this. Okay, interleukin 1 alpha, interleukin 1 beta, interleukin 6 and interleukin 8. Okay, 1 alpha, 1 beta, and CSF. Okay, G and M. CSF, interferon, TNF, TGF alpha beta, PDGF, FGF, MSH. So immunohistochemistry, 54 functional creatinines. Okay, type 1 to type 17, 27 genes. Type 1 and type 2. There are basically two types. Okay, type 1 17q 21.2, 12q 13.3. Not important. Both have 27 genes. You can write this. Okay. So K5, K14 basal cells mutations, epidermolysis bullosa, basal cells mutations. Okay. K1, there is a basal layer of the cells, basically same. So we have K1, K10 suprabasal. What is suprabasal? Spinous layer. Spinous layer we have bullous ichthyosiformis, erythroderma, epidermolysis hyperkeratosis. Then K2, terminal differentiate keratinocyte. This is a present corneal layer, corneum layer. Okay. K9 suprabasal keratinocytes in palmar and plantar epidermis. Mutation will cause epidermolytic palmoplantar keratoderma or Warner syndrome. Epithelial IHS marker P53 mutation will cause ectodermal dysplasia syndrome, overexpressed in SCC and BCC, squamous cell carcinoma, basal cell carcinoma. Okay. Then melanocytes, neural crest cells drive dendritic cells for the pigment synthesis. These contains melanosomes. On light microscopy, pale staining cytoplasm, ovoid nucleus, pigmented containing melanosomes present in basal layer extending to dermis, but above lamina densa. There is nothing much to understand here. Abdominal melanin unit is there here. I'll talk about it again. And it's basically what it's saying is what abdominal melanin is 1:36. For every 36 keratinocytes, there is one melanocyte. Racial difference of color is due to the varied distribution and size of melanosomes. What is a melanocyte? Just dendrites. This is the nucleus, rough endoplasmic reticulum, mitochondria, Golgi apparatus, melanosomes, melanin granules. Okay. So melanin synthesis is very important pathway. Tyrosine inhibitor as skin lightening agents, that is hydroquinone. This is what we give you, hydroquinone. First we'll talk about the cycle. Tyrosine, dopaquinone. Then here we have pathway for two. One is pheomelanin, one is eumelanin. So pheomelanin we have cysteine acting here. Then cysteine dopa, benzathiazine derivatives forming pheomelanin. And forming eumelanin is a very complex process. So leukodopachrome, then here dopachrome tautomerase acts here, forming only dopachrome. Glucose is removed. Then dihydroxyindole. Then tyrosinase related protein 1 forms quinones. Quinones form eumelanin. So what is typing? Many synthesizing melanoma within the melanocyte. Melanosomes are passed on to surrounding keratinocytes through several stages from 1 to 4. And we'll talk about stages later here. So these are stages of eumelanin melanosomes. Electronic images are, this is not very well in this book, but in this PPT, but try looking for it in your rooks. Okay, spherical, no melanin deposition. Okay. Spherical, no melanin deposition. Oval, obvious matrix in the form of parallel longitudinal filaments. See, parallel longitudinal filaments. Minimal deposition of melanin, highly tyrosinase activity. Oval and moderate deposition of melanin. See, it is less darker than this, but it is less darker than this, but more darker than this. So high tyrosinase activity. Here we have minimum tyrosinase activity. Same thing, high tyrosinase melanin. Just in order it is going right. From here we get oval. Here it is spherical. You can see here spherical, oval, oval. Okay, these are stages of melanosomes. These are 4 major stages. So melanosome transfer. Okay, one second. Okay, so melanosome transfer. Melanosomes are transferred from melanocytes perikaryon to melanocyte tips. Okay, through microtubules dynein and kinesin. Okay, RAB27 helps to bind melanosomes to myosin 5A via linker protein melanophilin glycylin synthesis. Autosomal recessive disorder with mutation in Griscelli syndrome. What am I talking? Griscelli syndrome. Autosomal recessive disorder with mutation in any of these above proteins. So, melanosome transmit across. See, we have dynein, kinesin, melanosome, RAB27, MLPH, MYOVA, actin. Applied aspects: albinism, freckles, nevi, melanoma, vitiligo. Effective formation of melanin: albinism, freckles, localized increase of melanin, nevi, benign proliferation of melanocytes, melanoma, malignant nevi. Okay, and then we have vitiligo, melanocytes are destroyed. So what is this? What is this? Albinism, freckles. Okay, what is this? Vitiligo. What is this? I will differentiate this. Which is nevi, which is melanoma. So irregular border is here, right? Irregular border. This is melanoma. This is nevi. Dendritic cells, dopa negative and ATPase positive. What are these? These are Langerhans cells. Dendritic cells, dopa negative and ATPase. This is, you remember, dopa negative and ATPase positive. Present in basal layers, spinous and granular cell layers. Pale stained with convoluted nuclei like melanocytes, but with no melanosomes or pre-melanosomes. Has special rod-like granules or the Burbeck granules. The tennis-shaped granules. Burbeck granules. So has special rod-like granules, the Burbeck granules. Function: antigen-presenting cells, stimulation of T cells responsible for phagocytosis. Okay, phagocytosis. Okay, like melanocytes but with no melanosomes. Has special rod-like granules for the Burbeck granules. Just tennis racket-shaped granules like this. Okay, these are all reading since your MBBS days, not required to revise. So we have the applied aspect. What are the applied aspects of Langerhans cells? We have psoriasis, sarcoidosis, conjunctivitis. UV rays impairs its function. Due to antigen presenting ability, these are vehicles for tumor vaccines or for tumor therapy. Merkel cells, slow adapting type 1 mechanoreceptors found both in hairy and glabrous skin of what is glabrous skin of non-hairy skin basically. Okay, digits, lips or oral cavity and outer root sheet of hair follicle. Pale staining cytoplasm, lobulated nucleus and cytoplasmic spines are towards the keratinocytes, seen in the basal layer forming desmosomal connections with keratinocytes. Immunohistochemical markers are K8, K18, K19, K20. Merkel cells. This you'll be reading in your previous days too. K8, K18, 19, 20. K20 is restricted to skin and the most reliable. Okay, has a role in Merkel cell, CA net, trabecular CA, but not well understood. What is this Merkel cell CA? Merkel cell carcinoma, net, trabecular CA. Intercellular junctions links the adjacent keratinocytes for mechanical biochemical signaling interaction between cells. Components: desmosomes, adherens junction, gap junction, tight junction. Desmosomes. Desmosomes. We have these regions are in the plasma membrane that connect between the cells and also tonofilaments with the cell membrane. Gives stability to epidermis. The core is rich in transmembrane Ca2 plus binding glycoprotein. It is desmoglins and desmocholine. There are three specific desmoglin 1 to 3 and desmocholine 1 to 3. And intercellular parts of desmocholine and desmoglin are attached to keratin network by desmoplakin, placoglobin, and other macromolecules. Adherens junction. Electron dense micro electron dense transmembrane structure that are associated with actin skeleton. Calcium dependent E-catenin is a component. The main linkage to actin is via alpha catenin. Here we have the structure again here. Catenins, plasma membrane, catenins, veniculins, alpha actinins. Okay, actinin, too much in depth, not required. But yes, you must have idea of what this image is. Gap junction comprises of series of intercellular connections known as connexons, allows the transfer of ions and small molecules between the cells. Okay, gap junctions. Okay, connexons, connexin. Tight junction, major regulatory epithelial permeability and skin barrier integrity, composed of occluding claudins and jam. Okay, occluding claudin, E-catenin, ZOM, jam, 1-catenin, singulin, actin. Epidermopoiesis. The epidermal thickness and the number and size of epidermal cells remain constant with the rate of cell production matching the rate of cell loss. Primary active. What is epidermopoiesis? This is definition of it. Primary active site for mitotically active cells are in stratum basal. Some suprabasal layer keratinocytes can also undergo cell division. Not important. Duration of cell cycle for normal epidermis varies from 50 to 44, 57 hours. In psoriasis, it is 36 hours. It is 57 hours. Mitotic division occurs in keratinocytes every 8 to 19 days. Time taken for keratinocytes to travel from basal to corneum and the skin surface is called epidermal turnover time, which is 50 to 75 days. Basal layer to stratum corneum, it takes 12 to 19 days. Stratum corneum to skin surface, 14 days. Do the math, you'll get this. So this is the mitosis cell cycle, basically. Not that's something new you're reading of. So double checks G2, double checks. This is one thing that you can remember. Double checks the duplicated chromosomes for error, making any need for repairs. Mitosis, cytokinesis, cellular continents, all this. Okay. The proliferation cells go through cell cycle, which is defined as time interval between two successive mitosis. The multiplying cells pass through 4 phases: the mitosis phase, followed by interphase, a post-mitotic phase, G1 phase, and active DNA synthesis. Okay. And we'll end our class from this last